Okay, in this video we will talk about some ways that we can incorporate environmental and habitat data in with our genetic data to give us some additional context for these types of dispersal and gene flow studies. So landscape genetics is really interesting and unique because it combines this population genetic data with environmental data to determine the actual geographic forces that are influencing gene flow across a landscape. So this is an image of some wetlands interspersed in spruce forest. So landscape genetics would try and figure out, okay, we have these different environments with the wetlands. Are the wetlands preventing gene flow for some species? Are they helping gene flow for other species? But the idea here in landscape genetics is we're focusing not only on the gene flow itself, but what actual processes are affecting it. And the overall goal here of landscape genetics is to understand the relationship between gene flow and the habitats. And when we're talking about habitat, that can be either natural or man-made habitat features. So this is one really cool example, mostly because I think these geckos are super cool. They're leaf-tailed geckos in Madagascar. I did not know these things existed until I found this paper. Look at that guy, his camouflage is top notch. In this paper, what they did, and this also ties in really nicely with what we talked about previously with our phylogeography topic. So what they did is collect geckos from different locations in Madagascar, and first of all what they did is build a haplotype network, which is shown here, which we all know how to interpret and even make at this point. So they had three different populations, A, B, and C. Based on this haplotype network here, are those three different populations genetically similar or genetically different? Right, we would say that they are genetically different because all of their haplotypes are unique. There is no haplotype that is shared for multiple populations. We also see very large separation here. Remember, each hash mark indicates a genetic difference, a nucleotide change. So population B has many nucleotide differences at this haplotype from the other populations. So overall, this haplotype network is showing very clearly that individuals from these three different sites are genetically differentiated. Where a landscape genetics approach comes in is not only did they construct this haplotype network, but they also then tried to look at the physical habitat features of these sampling sites and see what kind of processes might be explaining or contributing to this genetic pattern. And what you can see is that there are several different possible barriers that could be affecting dispersal and gene flow for this little leaf-tailed gecko. So in this part B here, we see our sites for A, B, and C. We also see some of the topological or man-made features that are separating these sites. So in black here, this is the national road. So it could be that they're similar to what we talked about in the overview with the highways. There could be a road that's causing a barrier for dispersal and gene flow. This river could also be affecting dispersal and gene flow for this species. So this is a nice example integrating previous topics and also illustrating this point of how we're trying to figure out, based on the habitat features, how that might be influencing the genetic patterns of differentiation. So one common pattern that can be found when we're doing these types of landscape genetics approaches is called isolation by distance, or that's often abbreviated IBD. And this type of pattern occurs when populations that are geographically close to each other have high levels of genetic similarity, while more distant populations are less genetically similar. So if we again use our hypothetical example that's supposed to be California, if we have population 1, 2, and 3, an isolation by distance pattern would occur if these two populations are relatively similar to each other, but these two are not. Again, that would be representative of this pattern that geographically close to each other are more genetically similar, presumably because it's easier to have gene flow occur between these two populations, whereas more distant populations are less genetically similar or more genetically different because of a lower occurrence of gene flow. So when we are trying to figure out if a pattern of isolation by distance is occurring, it's often decided using something called a Mantle Test. So we do this statistically. This is from a paper looking at different types of plants in China, diploid plant species. And the way that we set up these tests is we create scatter plots where on one axis is geographic distance, so this is our physical distance on a map, and then the y-axis is the genetic difference, so that's often the proxy for genetic difference is often FST, like we've talked about before. And so what we're looking for in the scatter plots is seeing whether there's a significant correlation. So like we said, with isolation by distance, the more genetically distant you are, the more geographically distant you are as well. So what we're trying to do once we plot all these data points for all of these different sites is see if there's a significant correlation here. In this case, the R-squared value was relatively high, 0.6, and the p-value was indeed significant. So in this case, we would say for these plants, isolation by distance is indeed occurring. Landscape genomics is a recent extension of landscape genetics and is taking this one step further in the sense that we're still looking at environmental factors, but now what we're focusing on specifically are the adaptive genes that help increase fitness in these unique environmental conditions. This is what I think is always so interesting. This is one of the reasons I was drawn to molecular ecology, is thinking about this connection between these adaptive genes and why they're able to survive in these unique habitats. There's a really cool example of landscape genomics related to oak trees in your textbook, but I always like to try and use previous examples and extend on previous examples to help us all make connections between what we've talked about before and what we're talking about now. And a really nice example of a landscape genomics approach that we've talked about before is these different acropora corals on Ofu Island in Samoa. And previously, we talked about this as an example of balancing selection, where in these two different pools, highly variable and moderately variable that differ in temperature, the different alleles are maintained in these different pools through balancing selection. This is an example of landscape genomics because we're incorporating environmental factors. So the environmental factor that we're looking at here is temperature. And then we're also looking specifically at adaptive genes. So if you look in this paper, which I'll put on Canvas, they specifically pulled out the outlier adaptive genes. And in this part A of the figure, which I had previously omitted, but now I'll add it back in since it's relevant, what they are showing here is that for one particular outlier locus, they have time above a certain temperature that is inducing heat stress for these corals. So time above 31 degrees C is on the y-axis, and the different genotypes are on the x-axis. And what we can see is that the BB genotype, so if you have two B alleles, that is much more commonly found when you spend more time at high temperatures. So this is an example of a landscape genomics approach. We're integrating that environmental condition of temperature with different alleles that are present at the unique adaptive genes. And when we're taking these type of landscape genomics approaches, often the type of thing that we're looking at is for something called a genotype environment association, or GEA. And in these types of approaches, what we're doing is asking whether selection can explain why some genotypes are more beneficial than others in these unique habitats. So just like we talked about with that previous coral example. And when we do have these types of connections between genotypes and environments, often we'll see not a pattern of isolation by distance, which is what we talked about before, but instead a pattern called isolation by environment, or IBE. And this occurs when populations that inhabit similar types of environments are more genetically similar to each other. So here the main factor is not the geographic distance, but rather the conditions of the habitat and the environment. So when we're saying similar types of environments, we can talk about either abiotic or biotic factors. So biotic factors could be something like presence of certain predators or prey. Abiotic, like we talked about before, temperature, precipitation, humidity, all that kind of thing. So there are many different types of possible variables we can investigate in the habitat. And for these IBE approaches, when we're thinking about what type of markers to use to determine whether populations are genetically most similar to each other, that's where we're using those outlier loci, which we've talked about before. So again, IBE approaches focus specifically on those outlier loci. So if we return to this frequency distribution plot, if you're using an IBE approach, you're only focusing on those outliers here that have an unusually high level of genetic differentiation compared to the SNPs overall throughout the genome. And what we can do by comparing the distribution of outlier loci with these different environmental variables is figure out if certain alleles under selection are associated with particular environments. So say we have one particular outlier locus, 5740. Remember, we had those similar types of designations in that lab we did for genetic diversity. So say at this one particular outlier locus, we have two different possible nucleotides. We have either an A or a C. If we find that this particular allele is predominantly found in warm environments, whereas this particular allele is more commonly found in cool environments, again, this could be something related to precipitation or something like that. If we take this type of approach, it allows us to see if certain alleles are associated with these unique environments. So that's the overall idea of these IBE approaches. A really cool example of this from a recent paper is one that they did dd-rad or rad sequencing on different individuals of Yucatan jays in the Yucatan Peninsula. And what they did is differentiate, like we've talked about previously, using neutral SNPs versus adaptive SNPs. So when they only looked at the neutral SNPs, they found that those SNPs followed a pattern of isolation by distance, meaning populations that were more geographically distant from each other were more genetically different from each other. But when you look at the adaptive SNPs, which we talked about is the focus for IBE, those adaptive subset of SNPs specifically follow isolation by environment patterns. So in this image here on the right, we're showing where specifically the Yucatan is, where the study was performed. These are the Yucatan jays that were studied. And then on the very far right is an image of the sampling, a zoom in of the sampling region. And these birds occupy these different types of habitats. So some of it is tropical dry forest, some is tropical subdeciduous or tropical evergreen. And so specifically when they were looking at those adaptive SNPs, they found that temperature, in particular temperature and precipitation, appeared to be among the main drivers of adaptation along the environmental gradient here, specifically for those adaptive outlier SNPs. One last example that I wanted to talk about for a couple different reasons. I'll explain my reasons. This is a study where previously using microsatellite loci, they had found evidence for a panmictic eel, quote-unquote, superpopulation. So when I think of eel superpopulation, I think of the Princess Bride with the shrieking eels. I like that movie. It's one of my favorite movies. I watched it again recently, so I wanted to throw in that little reference there. That's the first reason I wanted to use this example. The second reason is that it's a really cool example and illustrates one of, like I've said over and over again, my favorite things where we're connecting specific genes and adaptive SNPs to environmental differences that kind of make sense in terms of why that particular gene is helping them in that particular environment. So previous studies using microsatellites had found that these eels across different locations were all genetically similar. They were a superpopulation. But in 2015, they looked at these same populations again. I should mention these are American eels, so they're on the east coast of North America. They looked at these same populations again with rad-seq, and when they looked specifically at the outlier SNPs, they did find some differentiation. So outlier SNPs do not support this idea of a superpopulation, but instead does show some differentiation. And in particular, those outlier SNPs were associated with genes that likely contribute to phenotype differences. So in these particular eels, they're found in different environments, either freshwater or brackish saltwater habitats. And in those different habitats, the eels, even though they're the same species, have these drastically different phenotypes, which are shown here. Some of them are a lot bigger, some of them are a lot smaller, and they have differences in growth and that kind of thing. When they looked at these different populations with the different phenotypes, they actually found that a lot of those outlier SNPs are associated with genes involved in functions that make sense. Things like vascular and morphological development, calcium ion regulation, growth, olfactory receptors. And so all of these SNPs that they found were outliers to have these strong genetic differences between these different phenotypes are likely things that are helping these eels adapt to their different freshwater or brackish saltwater habitats. So that's an example of how we can integrate this genetic data, genomic data, with environmental data to take this landscape genomics, isolation by environment type approach.